All right, so this is our first lecture. As I've indicated previously on my emails on Blackboard, I'm going to try to add some audio to the lectures so you guys have something to sort of complement what you're doing in terms of your reading and your notes and all that kind of stuff to make sure you're understanding stuff. So anyway, this first chapter, this first unit that we'll be doing actually is the cardiovascular system. And the cardiovascular system, as an organ system, has three components. It has blood, of course, which is what we're going to be doing today. It's a fluid medium. It basically has, a fluid matrix that has cells within that fluid matrix. It's about half water, roughly half cells, mostly red blood cells. So that's one part, which is what we'll look at today. Other part is the heart, which we'll do next, which is a pump, basically. You guys know what the heart is. It's a pump. It squeezes and pushes the blood throughout the system. And then you need some sort of conducting system to carry that blood to, all the different organs in our body so they can survive and get the blood with the oxygen and the nutrients that contains that. So, so that's what the cardiovascular system is, and that's what we're going to do in this first unit here. Correspondingly, we'll be doing similar things in lab, like some blood exercises. We'll be doing a heart dissection, or I don't know if we'll do a heart dissection because our lab may not be ready, but we're doing heart models and some other types of things like that. So, but that's what this first unit is going to be, and we're going to start with the blood. The blood is, very important in terms of a functional standpoint. Basically, it transports materials to different cells and organs, essentially, in our body, as I mentioned. So you have oxygen obviously being one of the most important things. You have red blood cells that have the ability to bind and carry oxygen throughout our bloodstream and arrive at tissues, and they can give off the oxygen. They can also take up carbon dioxide, which is often a waste product or a byproduct of aerobic metabolism and other things, and attach that and take it back to the lungs where it can be given off. Nutrients also travel in our blood to be delivered to targets. Hormones, as you recall from the endocrine system lecture last time, also travel in our blood. Immune system components, as we'll look at here, white blood cells. And then, of course, waste products. Our body uses blood to get rid of waste products, oftentimes getting rid of those things through the urine and the kidneys, which we'll talk about later on in this semester. Okay. Important functions of the blood, as I said. One of the big things, obviously, is transportation, right? As the things we just all talked about, oxygen, CO2, hormones, nutrients, all those things are basically transported, essentially, in the blood to the target. But also one thing that's really important, as we're going to see, is regulation of pH. That's something that's really important. Restriction of fluid losses at injury sites. Body temperature is one thing that's really important. So we'll look at all those components when we, as we sort of progress through here, okay? All right, so what is blood made of? Well, there's basically two things primarily, two major components. You have plasma, which is one thing, and then you have formed elements, which is another part, which is basically plasma is primarily water, and formed elements are primarily cells. Okay, and so we want to take a look and see, what those components are and what they're made of as we sort of go through this. All right, move along here. So here's a nice image sort of showing what I was just talking about. The plasma, as you can see, 55%, the vast majority of that is water. The other component, which is formed elements, typically about 45%, and most of that comes from red blood cells. As you can see, white blood cells and platelets make up a very small volume. So the vast majority, as you can see, is water and red blood cells. That's pretty much what your blood is. There are some other things like solutes and plasma proteins in the plasma that we'll talk about, but those are the primary components. So some physical characteristics of blood. Hemopoiesis, is how the formed elements are manufactured, essentially. We'll look, they come from essentially what are called myeloid or lymphoid stem cells that are going to produce red blood cells and white blood cells, essentially, and platelets. Those are all the formed elements. And Those of you who have had experience with blood may have seen this or worked with it, where you can actually separate whole blood for analysis. It's called fractionation, where you take like a centrifuge and it spins really fast, and the force of that spin with gravity puts all the formed things towards the bottom and puts all the water and fluid stuff at the top. And so that's how you can separate it into plasma and formed elements so you can analyze the blood. All right, so as I said, the formed elements consist of 99.9% red blood cells, or RBCs, also referred to as erythrocytes, as we talked about. Transporting oxygen is something that's really important. White blood cells, or leukocytes, which are part of the immune system. We'll talk about their function. You guys get an opportunity to look at some white blood cells in lab. And then platelets are the last sort of component of the formed element part. And they're essentially cell fragments that are involved in clotting, right? So if you have an injury of some kind and you have platelets that aggregate and they form a plug, essentially where the opening is, ultimately that's going to start to get stronger and stronger. As we remember from ANP1, it's a positive feedback loop, and you get a clot. So that's what the platelets do. So those are the three parts of the formed elements: red blood cells, white blood cells, and platelets. Okay. Some characteristics of blood: it's a little bit warmer than normal temperature, 100.4. Has a pretty high viscosity. You guys know what viscosity means, right? It's sort of a thickness, because it's not just pure water that has formed elements in there as well. So it's going to give it a certain amount of thickness. And it's slightly alkaline, just above, pure water is a pH of 7, so 7.3, 7.4 roughly. That is the pH of blood, so it's slightly basic. Any change in that pH can be very toxic and dangerous, so that's one of the big jobs of the body, is to maintain pH. And so we'll look at that later on in the semester when we look at the kidneys and how they help regulate pH balance and things like that. All right. Blood volume, I think you guys can look at that. Composition of the plasma, look at the plasma first. As we said, more than half of your blood is plasma, okay? And more than 90% of that is water, okay? So it's water mostly, but it has ions and solutes and other nutrients and other types of things floating in the plasma as well. The plasma proteins, which make up about 7% of the plasma, consist of three primary types of plasma proteins: albumins, globulins, and fibrinogen. So you can see the albumins are really 60%, so that's a big portion of that. So albumins oftentimes are transport proteins. You guys, we saw these back when we looked at the endocrine system, like thyroid hormones needed a transport protein to sort of carry it throughout the body. Certain steroid hormones needed transport proteins as well. And so those primarily comes from this collectively things known as albumins, which again are 60% of the plasma. Globulins are 35%. Those are primarily antibodies. We'll talk about antibody production later on when we do the immune system here in a few chapters. And then finally, fibrinogen is the last component, as we'll see coming up here in a little bit. All right. So other plasma proteins, things like hormones, for instance, are found. Peptide hormones typically are circulating in the blood. That's just going to make up about 1%. So insulin and glycoproteins and TSH, FSH, things we talked about last time in the endocrine system are all found there as well. Plasma proteins, most of them are coming from the liver, actually. The antibodies, as we'll see when we do the immune system, come from plasma cells, which are part of our acquired immunity. And then, of course, the peptide hormones are coming from the actual endocrine organs, right? Like the pituitary gland is producing TSH, and so, that's where they're coming from, the actual endocrine organs. So that's the location of those. Red blood cells, as we said, making up 99.9% of the blood's formed elements. Hemoglobin is the red pigment that gives blood its color, right? So that's why when we look at arteries, we typically color like arteries and show them as red, because they're bound to oxygen typically in arteries, and so it gives them more of a reddish color, whereas we show veins typically in blue. You guys may know that, but again, because they're deoxygenated, so we show that in blue, because the oxygen is what's giving blood its color, because this hemoglobin is in the red blood cells, as we'll talk about in a minute. And this hemoglobin not only does it give it blood its color, but it also is what has the ability to bind and transport oxygen and CO2. Okay, so those are important functions that we want to look at. I think you guys can read that okay in terms of the abundance of red blood cells. Hematocrit is something that gives you what's called packed cell volume, or PCV, which is the percentage of red blood cells in whole blood. We can do blood tests and other types of things to get hematocrits easier, but the old school way of measuring hematocrit was taking a sample of whole blood, as I said, putting it in the centrifuge, spinning it really fast, and you separate it, right? You separate the plasmas on top, they're packed components on the bottom, and whatever percentage of that, you can measure it. They used to have like a little card that you can hold the tube up to and measure. That gives you your percentage of formed elements in the blood. And so typically the formed elements is, somewhere in the 40% range. Typically, it can be a little bit different between males and females. It can be a little bit different depending on your health status and how fit you are. Other types of things like that can impact it. But that's what hematocrit refers to, is the percentage of red blood cells in a whole blood sample. So, structure of red blood cells, really important to understand the structure. They're very unique. They're very small, of course, but they're specialized discs. They're actually thin in the middle, and they get thicker at the edge. So if we look at an image here, you can see what I'm talking about, right? They're thin here in the center, and then they get larger as they go to the outside. Okay? And this shape is really significant and important in terms of what blood is able to do. Okay, and so these three important effects of red blood shape on function, really important slide here. High surface to volume ratio. What does that mean? Well, it just means because of the shape, you can quickly absorb and release oxygen, because you want to be able to bind the oxygen, but you want to be able to release it very well, or very easily as well. So that's important. The shape of these discs, when they get thin in the middle and thicker on the ends, gives them the ability to form like little stacks, and we call that rouleau, which basically it's almost like stacking plates in your cupboard or something like that. They're able to fit right on top of each other. And the reason that's so significant and important is because not all your blood vessels are exactly the same size, and certain blood vessels, specifically capillaries, get really, really small. You can have microscopic little capillaries, and so these red blood cells need to get down, obviously, to those capillaries, because that's where the exchange of the gases is occurring. And so you need to be able to stack these up so they can move and smooth the flow of blood through those really narrow vessels. Okay, so that's important to form rouleau, and you can see that. I have an image of that somewhere right here, right? See that? See how that looks like rouleau, right? See how they're formed little stacks? Here's another nice little section. So this is like a section of a capillary, right? And so they need to be able to go into that location. Last thing that's really important about the shape and the sort of structure of these red blood cells is that they have the ability to bend and flex. So they're pliable. They're not like stiff as boards or something like that, right? Because again, entering these really small capillaries where they're really narrow, 4 micrometers, right? You're going to need a little give and take there. They can't just be rigid as they go through these really small areas. So these things are all really important in terms of what they give to the functionality. The shape or the form follows the function and what red blood cells are capable of doing. So this is a blood smear. You guys are going to get a chance to do this in the lab. You can see how they're kind of, these red blood cells are shown. You can see how they're kind of thin in the middle and then thicker on the outside as you look at all those different red blood cells. And here's an actual 3D image. Again, you can see how they sort of taper in the middle. Here's a nice little stack of rouleau, as we were talking about right there, of red blood cells. All right, and then I already went over this through the narrow plates. All right, lifespan of red blood cells. There's no nuclei or mitochondria or ribosomes, any of that stuff that you typically see in normal, sort of garden variety cells. So no repair. And it's going to use anaerobic metabolism, right, because there's no mitochondria. So they're going to live about 120 days, and so they go through this life cycle essentially where they're recycled essentially. Once they're done, their lifespan is ended, the body has the ability to recycle and reuse certain components, okay? And we'll talk about that in a minute. Big part of red blood cells is this hemoglobin that I mentioned before. This is what gives it color, but it also has the ability to bind and transport these gases, okay? So you can see the normal levels in the adult male and the adult female, fairly similar in terms of the amount of hemoglobin in a gram, 12 to 16 grams per deciliter for women and 14 to 18 grams per deciliter for men in terms of what you typically see. The structure is very unique. It's this complex quaternary structure that has 4 globular protein subunits, each with one molecule of what's called heme. Heme is attached to one iron ion. Okay? And so if you look at this image, I think it'll help you see that. Okay? Here is, they're just sort of pulling out one hemoglobin molecule from a red blood cell, right? And you can see there's 1, 2, 3, 4, right? Alpha chains, beta chains. And if you look at each one has a heme molecule essentially. And if you look at one heme molecule, you can see it has one iron ion in the center. Okay? Iron ions are really important because they have the ability to associate or attach or bind with oxygen, of course, but also disassociate easily from oxygen, right? You want to be able to bind the oxygen at the lungs, right? When you breathe in, you pull that oxygen in, it binds and attaches to the hemoglobin, right, forming what's called oxyhemoglobin or HbO2. It then travels through the blood, gets to the target where there's less oxygen and disassociates very easily, and then the process, of course, just continues to cycle in that fashion. All right? So that is the structure of hemoglobin. Now there is what's called fetal hemoglobin. You guys may have heard about this, or at least the concept of how a baby can get oxygen from the mother. And actually the hemoglobin in a fetus, as it's developing, has a stronger affinity for oxygen than adult hemoglobin does. And so it binds oxygen more readily, and therefore the developing fetus ensures that it's stealing or getting enough oxygen from the mother's hemoglobin. And then ultimately after birth and for about a year, gradually the newborn converts to adult hemoglobin. Okay? All right, so hemoglobin function, again, it carries oxygen. The hemoglobin, of course, releases oxygen, but also has the ability to bind carbon dioxide. So as we'll see when we look at gas exchange a little bit later when we do respiration, we'll see that when, red blood cells carrying oxygen, say, are delivered to like your muscle, for instance, or your brain or wherever it's going, it's going to disassociate with the oxygen, but it's also going to pick up some CO2. So this process, this trade-off essentially happens and forms what's called carbon-amino hemoglobin, right? It's going to bind that carbon dioxide and then carry it back to the lungs where it can be released, oxygen is gained, and that's the cycle that it goes through. Okay? One clinical example of a dysfunction or disease is called sickle cell anemia that you may have heard of. Basically results from a mutation of the hemoglobin, specifically the beta chain. At low oxygen levels, the red blood cells become sort of rigid and sharply curved as a result of this mutation, and makes them have the ability of sickling or damaging or cutting or, injuring adjacent red blood cells. And so these malformed cells, not only do they become problematic, but they can injure other cells, and you can potentially, these cells can get stuck and cause a circulatory blockage, and so that's why it can be so dangerous. Okay? So if you look at this image, here you can see some normal red blood cells, but then you can see some sickled cells as well, and you can see how they kind of have like a sharp point on them and really abnormal looking red blood cells. Okay? So that can be dangerous and cause clotting issues. All right, so red blood cell formation, as I said, and turnover, you get a pretty fair amount of turnover. 1% of red blood cells wear out each day. So that means you're going to have to create and manufacture and replace that new red blood cells every second. So it's a pretty significant process. So we'll take a look here at the formation of red blood cells, and then how also when you recycle, what you can do with the hemoglobin, how you can recycle and reutilize it, primarily using macrophages that sort of monitor and engulf these red blood cells before what's hemolysis occurs, which is when the actual cell itself ruptures, releasing all of its content. So we'll look at how that process works. All right, so the process basically, when a cell has lived its 120 days and it's basically time for it to be recycled, phagocytes basically break down the red blood cell and then break the hemoglobin actually into its components, right? It's primarily made of amino acids, heme, and iron. Those are the three primary components. The globular proteins can be converted to amino acids. The heme is going to be broken down to what we call biliverdin, which will then go into what's called, not the greatest drawer here yet, we'll go to bilirubin, and then we'll see how that's processed. And then also you have the iron that can be reused as well. Okay. This is just what happens when you have excessive breakdown of products. For instance, if you have a lot of hemolysis, like maybe if you got a blood transfusion of the wrong type, you're going to get a lot of destruction of red blood cells. And so you're going to get excess hemolysis in the bloodstream, so your urine would probably look sort of reddish as you are breaking down rupturing all these red blood cells. Hematuria is whole blood cells in the urine due to kidney or tissue damage because they're not working properly. All right, so the one component that I mentioned, biliverdin, is a byproduct of breaking down the heme, and biliverdin is has sort of a greenish pigment, is converted to what's called bilirubin, which then has sort of a yellowish color. Bilirubin then is typically excreted in bile, which is manufactured by the liver. And so we talked about jaundice last time, and this is really where it's coming from, right? It's the liver and processes and produces bile from this bilirubin, and if it's not handled correctly, it can spill over into the tissues, and so you can get jaundice, which is a buildup of bilirubin, and that can give you sort of a color, yellowish, orangish hue to the skin, and we talked a little bit about that last time in AMP1. Iron pretty much can be recycled. It uses transferrin as a transport protein to take it back where it can be reutilized in forming more red blood cells. And so this slide, you're going to probably need to either look in the text or on the actual PowerPoint that I have. But you can see this sort of shows all those processes in terms of red blood cell formation from bone marrow, right? And then the average lifespan is 120 days. You can see the different percentages of ones that are, vast majority of them are going to be broken down by macrophages. And you can see the different components. The heme, you're going to take the iron, transport that back to the red blood cell so it can be utilized again. The other part is going to be converted from biliverdin to bilirubin, which will be manufactured in bile and typically processed either in the digestive pathway or the kidneys. So make sure you review this in terms of understanding what's happening and how red blood cells are not only manufactured initially, but mostly recycling of these red blood cell components. All right, so what about red blood cell formation then, or manufacturing or production? This process, it goes through what's called erythropoiesis. Okay, erythropoiesis only occurs in myeloid tissue, which is red bone marrow, and ultimately they're going to convert into mature red blood cells. They start at what are called hemocytoblasts. They're stem cells that ultimately will convert into either myeloid stem cells or lymphoid stem cells. Myeloid stem cells pretty much manufacture every type of red blood cell except lymphocytes, which come from lymphoid stem cells. And I think I have an image here, where you can see that. Right here is a hemocytoblast. It's going to convert either to one of two things: a myeloid stem cell or a lymphoid stem cell. As you can see, myeloid stem cells basically produce just about everything, all of the from red blood cells to platelets to a lot of the white blood cells. The only one that's producing that isn't producing are lymphocytes. Lymphocytes are coming from lymphoid stem cells. So make sure you have a really good understanding. We'll come back to this slide many times here in lectures, so make sure you have a really good understanding of that process. All right? All right, so hemocytoblasts are the stem cell that ultimately is going to convert into either a myeloid or a lymphoid stem cell. Okay? And from that point, it goes from a myeloid stem cell to what's called a proerythroblast, then an erythroblast, a reticulocyte, and then finally it becomes a mature red blood cell. And then it's in the bloodstream and utilized for approximately 120 days. So here you can see that same process more in an illustration. Takes about a week or so, maybe 7 to 10 days to manufacture these red blood cells. So you can see proerythroblast here, right? Erythroblast, normal blast, and at this point you're seeing the ejection of the nucleus. Remember we said that these red blood cells do not have nuclei, okay? And then it's going to enter the bloodstream as a mature red blood cell, okay? All right, so how is red blood cell formation regulated? Well, first of all, you need several things in order to successfully build a red blood cell. Obviously, you need iron. We talked about that because that's, you know, what has the ability to bind oxygen, so you absolutely need the iron. You need the amino acids to make the components of the hemoglobin. And then there are certain vitamins that you need, B12, B6, folic acid, all really important in terms of formation. So that's one thing you absolutely have to have. And again, if you don't have it, like for instance unavailability of vitamin B12, you're going to see low red blood cell production. Okay? Now, what stimulates red blood cell formation? Well, we have a hormone, and we talked a little bit about this in AMP1 when I talked about cyclists and other athletes doping, blood doping, and things like that. We said they use a synthetic form of EPO to stimulate red blood cell formation. Well, the natural form of EPO is naturally occurring in our body. It comes from our kidneys. It's also referred to as erythropoiesis, stimulating hormone, which of course is going to stimulate erythropoiesis, which is the formation of red blood cells. Okay, it's typically secreted when you need more red blood cells, so either in disease or high altitude, or if you have a high turnover of red blood cells, you're going to get high levels of EPO being released. Okay, EPO stimulates red blood cell formation, and is released when red blood cells are needed. This shows just some of the red blood cell tests and terminology. So you might want to review these components so you can take a look at that. But red blood cells, you can see here's the abundance per microliter, what they look like, functions, remarks, etc. about red blood cells. All right, so that's pretty much it on red blood cells in terms of their structure and formation and how they're processed and regulated, and their lifespan and all that kind of stuff. Okay. Next, we want to take a look at blood typing. Blood typing is essentially, and I think a lot of you probably know this, you guys have all heard of like type A or O positive or whatever, the different blood types that you've probably heard of before. So we want to take a look and see what that means exactly. And basically what it means is all cell surfaces, the surfaces of all cells in our body have proteins on them. They call them surface antigens, which allows them to be recognized or identified by the immune system so that the immune system doesn't attack them essentially, right? So if you have what the immune system is looking for, it's an antigen, then it ignores it, right? The immune system doesn't attack it, right? That's how it is able to identify what it needs to attack based on either what chemicals it's giving off or what surface antigens are present. So in blood, you have these surface antigens that help identify our red blood cells and identify them as quote-unquote normal. And if they're normal, they're not going to get attacked, right? And so that's why it's so important, for instance, when someone has like a blood transfusion, right, that you make sure you have the match, right, in terms of the type of surface antigen or the blood type, right, or you're going to have a big issue where your body's going to attack all those red blood cells. You get a lot of hemolysis and other types of problems. So That's why blood typing is so important. So blood types are genetically determined, and basically it's either the presence or absence of a surface antigen. There are different ones. You have A, B, and RH essentially, and we'll sort of look at how that works. So based on these different antigens, you have 4 basic blood types: A, B, AB, or O. Okay, which is neither A or B on that surface antigen. Okay, and you have agglutinogens, antigens on the surface of the red blood cell that are screened by the immune system. Okay, if it's not the right antibody or antigen, on that surface, right? If it's not the right surface antigen, then your body is going to attack it. Plasma antibodies will attack and cause clumping or agglutination of those foreign components. Okay, so basic on what you have. If you have type A antigen, then you're going to have type B antibodies, meaning that if you get type B blood in your system, it's going to attack those, but it will ignore type A, right? And so same thing with type B, you have type A antibodies. Type O has both A and B antibodies, so that's why we call type O sort of a universal donor, and type AB has neither A nor B, right? And so this sort of shows you a nice illustration showing you how this works, right? You have a red blood cell here, it has surface antigens, and so if you have type A blood, then you have type A surface antigen. But if you have type A blood, you also have anti-B antibodies, right? And so in a scenario where you get type B blood, for instance, these guys will be able to attack and remove those. So type B has surface antigen Bs, of course, and then you have anti-A antibodies to attack A antibodies. AB, and then type O has neither, as I mentioned before. Okay, RH factor is another significant component to blood typing, and this is also referred to the D antigen, but instead of adding D to the type, we just say it's either positive for D or negative to D, and so that's why you have this plus or minus typically associated with your blood type as well. Okay, so we have RH positive or RH negative from that standpoint. And so as I said, you can get a cross-reaction in a transfusion, also called a transfusion reaction, where a plasma antibody meets a specific surface antigen that it doesn't like. It will agglutinate and hemolyze that blood, and will occur if it does not receive a compatible blood type, right? And so, and I worked at WashU, and I've talked about these things before, but we worked in the neuro ICU and worked with stroke patients, and oftentimes they would need a blood transfusion. And so obviously it goes without saying there were several steps involved to ensure that the patient was getting the proper type of blood for that transfusion. All right? So I think you guys all know about that. And so here sort of shows what happens, right? You have, in a cross-reaction, antibodies react with their targets, boom, and you get a lot of hemolysis and destruction of those red blood cells. All right, so again, without a cross-match, type O negative is typically a universal donor. So if you have that type of blood, you're probably going to be getting a lot of calls from the Red Cross and whatnot to donate, because it's a very universal type of blood type. All right, so this is just a slide that you don't need to memorize this or anything, but it gives you an idea of sort of the average types, percentages with each blood type. So as you can see, a lot of people have O-type blood. It's very common, right? Much more than, say, AB or even B-type. You don't see as many of those. Most you see O and A, and most are RH positive, right? So you don't see too many of the other varieties. But that just gives you sort of an idea of the difference of different blood types in our society. All right, so that's it on red blood cells. We can now look to the other sort of formed element, which is white blood cells. White blood cells, also referred to as leukocytes. They do not have hemoglobin, so these guys are not interested in carrying red blood cells and things like that. They do have nuclei and stuff, but basically what white blood cells are designed to do is defense. They're really sort of part of our defense and immunity. defend against pathogens. They can remove toxins and waste. We saw how they can also have the ability to, certain ones can help with recycling blood, and they can attack abnormal blood cells, other abnormal cells. So really quite a bit of function. So this what we look at with white blood cells today will primarily be looking at it from just sort of a manufacturing of white blood cells and how they're produced. We'll look more at immunity later on in the semester when we do that specific chapter. All right, so as you probably know, most white blood cells are not in the blood, actually, right? And if you do have like if you did a blood count or a blood analysis and you had high quantities of white blood cells in your blood, that would be dangerous. That would be a sign of an infection, or even worse, maybe some sort of disease or cancer or something like that. So typically, the amount of white blood cells in your blood are very low. Most white blood cells are actually in connective tissue. Remember we talked about areolar connective tissue back in AAP1 and how it has a lot of these white blood cells migrate out of the bloodstream and move into the connective tissue. But then they're also, of course, going to go into the lymphatic system and the lymphatic organs and lymph nodes and things like that, which again we'll look at more when we do immunity here coming up. So there are very small numbers in the blood, only 5,000 to 10,000 per microliter. You need to save all that blood space for plasma and red blood cells, right? So you don't want white blood cells crowding those out, okay? So some characteristics of these circulating white blood cells is they can migrate out of the bloodstream, as I just mentioned. They are able to move. They have what's called, can go through what's called positive chemotaxis, where they're attracted to certain chemical stimuli, which is how oftentimes they find the target that they want to eradicate. And some have phagocytic capabilities, meaning they can consume, damaged cells or abnormal cells, things like that. So those will be done by neutrophils, eosinophils, and monocytes, which we'll talk about in a minute. But quickly, just looking through this, just make sure you have a good understanding of how they have the ability to migrate out of the bloodstream, looking at this process called margination, where they actually interact with the endothelial wall and move in and out of the bloodstream to the connective tissue or to the lymphoid system. They have a certain level of mobility, which we call amoeboid movement. They are attracted, as I mentioned, to chemical stimuli, right? It's called positive chemotaxis, which guides these white blood cells to pathogens and damaged tissues and things like that, right? Positive chemotaxis. And some are phagocytic, as I mentioned. They can engulf pathogens, debris, and other materials. All right, so understanding sort of the function of white blood cells and really how it's quite different from what we're seeing with red blood cells. White blood cells, things like neutrophils, eosinophils, basophils, monocytes, and lymphocytes are the 5 types of white blood cells. And again, as I said, in lab, we're going to have the opportunity to sort of work through this and take a look at these under the microscope. Okay? As you can see, 4 of the 5 are coming from myeloid stem cells, right? There are 4 of the 5 white blood cells. As we said before, lymphocytes are coming only from lymphoid stem cells in terms of their manufacturing and formation. And here's a nice image of what these white blood cells look like. Okay, you can see here's what's called a neutrophil, has sort of this multi-lobed nuclei. Eosinophil has sort of a palish or reddish cytoplasm. Basophils have a darker cytoplasm with a multi-lobed nuclei. Monocytes are really large, and they have a very large nuclei. And lymphocytes are very distinctive as well, as they don't have as much cytoplasm, but a really large nucleus. Okay? So, talking about each one of these, neutrophils typically are about 50 to 70% of white blood cells. They have a paler cytoplasm, as I mentioned, has lysosomal enzymes and other types of things that can cause destruction and damage to abnormal cells. They're very active. They're typically the first to attack or arrive at a bacterial site or infections. As I said, they can engulf and digest pathogen. They can go through what's called degranulations to attack, and we'll look again at neutrophil action even in further detail when we do the immune system. Okay, and they can also form pus when they, damage these different cells that they're attacking. And so there's what a neutrophil looks like. Again, you're going to get the opportunity to look at these neutrophils in lab coming up. All right, eosinophils These are only 2 to 4% of the white blood cells. Typically they go for larger parasites. They have toxic components that can be released that are cytotoxic or kill off cells. Also very sensitive to allergens, so they can be involved in the process of inflammation as well. And so there's what an eosinophil looks like. As I said, it sort of has maybe a pinkish or lighter colored cytoplasm. Basophils, less than 1%, so you're not going to see too many of these. They can accumulate in damaged tissue and again release histamine and heparin. We talked about that back in AMP1 when we saw these basophils in areolar connective tissue. So those are some functions there. And there's a basophil in terms of its appearance. Monocytes are a little bit, see more of those, 2 to 8%. These again are very large, a little more spherical in shape. They can ultimately turn into what are called macrophages. Macrophages, if you recall, have the ability to engulf and consume large particles and pathogens, and they also contribute to positive chemotaxis by releasing chemicals and substances that attract further immune response and fibroblasts and other types of things to the area. So those are monocytes, right? Much larger cell, sort of kidney bean looking shaped nucleus from that standpoint. And then finally, the last of the 5 white blood cells are lymphocytes. Lymphocytes are 20 to 40% of what you're going to see in the white blood cells. They're a little bit larger than the red blood cells. Again, they can migrate in and out of blood. Again, most are in connective tissue or the lymphatic system and are part of what's the body's specific cellular defense system, which we'll talk about when we look at immunity a little bit later on. And there is a lymphocyte. Okay, so for now, I just want you to have sort of a general snapshot of what's happening with all of these 5 different white blood cells. And then specifically to lab, you need to be able to identify them, of course, and get a feel for what they look like and the percentages and all that kind of stuff. Now, lymphocytes, there are actually 3 different types of lymphocytes. You have what are called T-cell lymphocytes, which, again, we're going to go into much greater detail on this. I just want you to have a general snapshot of what lymphocytes are. But you have T-cells, which perform what's called cell-mediated immunity, in that they attack cells directly. Okay, and they're called T-cells because typically they get their, what we call their education, so they identify the correct things in the thymus. And so we'll talk about that a little bit later on. B cells are also a different class of lymphocytes. They're responsible for what's called humoral immunity, or immunity that we develop over time. They are able to differentiate into what are called plasma cells, and plasma cells synthesize antibodies, and antibodies then perform what's called antibody-mediated immunity. So you have cell-mediated immunity in the T cells, and you have antibody-mediated immunity in the B cells. And then finally, you also have a third class called natural killer cells that perform what's called immunological surveillance, where they survey and detect and destroy abnormal tissues. Oftentimes cancer cells can be the target of natural killer cells. Okay? All right, so as I said, if you have a abnormal differential count and you see an increase in white blood cell profile in your blood, that's going to be the sign of problem, right? Infection, inflammation, allergic reaction, but even something like cancer cells or things like that can be sensitive to higher levels of white blood cell count. So if you have high levels of white blood cell, you're going to have a disorder. You can actually have very low white blood cell count. That's called leukopenia. A normal, a little bit high white blood cell, like if you're having an infection or something like that, is leukocytosis. But if you have an extremely high white blood cell count, you probably have what's called leukemia. And leukemia, you guys are probably familiar, is a form of cancer or cancer of these white blood cells. And so basically what happens is typically you have like a genetic mutation of some kind where the blood cells are essentially manufactured abnormally. And not only are the white blood cells abnormal, but they produce too many of them. And that's sort of the hallmark of cancer, as we've talked about before, like with epithelial types of cancers like colon cancer or ovarian cancer, right? This epithelial lining starts to manufacture these abnormal cells and overproduce them. And so same thing happens here, except it's white blood cells that become abnormal and overproduced. And these leukemia cells don't function like normal white blood cells. They grow faster. And the big danger is they're going to crowd out the other normal blood cells, like red blood cells, that need to be there. And so they're going to cause a lot of problems. So if you look at it under the microscope, as you can see, under normal blood, if you're looking under the microscope, you're going to see mostly white blood cells. You'll see an occasional mostly red blood cells. You're going to see an occasional white blood cells and platelets here and there, but that's typically what you're going to see in a normal blood smear. In leukemia, right, look at all those white blood cells everywhere. Okay, they're causing all kinds of problems. So anyway, we're going to look at this a little bit further here real quick. Okay, so leukemia is generally categorized as either acute or chronic, or lymphocytic or myeloid. Acute or chronic means, you guys know what that means. Acute is immediate, right, in terms of its manifestation, whereas chronic is sort of a slower and longer progression over time. Lymphocytic or myeloid just really determines what cell is being impacted, right? We said lymphocytic are impacting lymphocytes, and myeloid are going to impact myelocytes ultimately. So from that, you can get 4 primary types of leukemia: ALL, AML, CLL, and CML. So acute lymphocytic leukemia, acute myeloid leukemia, chronic lymphocytic, and chronic myeloid. So those are your four types. And ALL is a pretty common one, so I thought instead of going into all these different types, I thought I'd focus in on one of these types of leukemia, and that's called ALL, or acute lymphocytic leukemia. And again, it comes from lymphoid stem cells that are turning cancerous. You're seeing about 4,000 cases a year in children, so it's the number 2 most common type of childhood cancer. It's more common in children, but also, for whatever reason, more treatable in children. You have a much higher survival rate in children that have ALL as opposed to an adult who gets ALL. It's very lethal in adults. Survival rate is only about 40%, and even less if it relapses. So that's sort of the significance of ALL. So what happens with ALL? Well, you're going to see swollen lymph nodes, increase in white blood cell, decrease in red blood cell counts, of course, as we mentioned before. Treatments, chemotherapy, maybe a bone marrow transplant, possibly if in severe relapse. Genetic sequencing is something that's starting to draw a lot of interest and use. And when doing my research on ALL and coming across this, I learned of this case of the WashU leukemia researcher named Dr. Lucas Wortman, and he is a leukemia researcher here at WashU currently, and recently he got ALL, got leukemia. So what are the chances of someone who is a leukemia doctor also getting leukemia? And as an adult, as I said, it's very dangerous and very difficult to recover from. So anyway, the link wasn't captured in this audio presentation, so you're going to need to go back to the original PowerPoint itself and click on this Dr. Lucas Wortman, and there is an article with a video about Dr. Wortman and about ALL and how they treated it, and it's very interesting. So go ahead and make sure you watch and read about Dr. Lucas Wortman and its association with ALL. All right? All right, so white blood cell production, again, we already saw they produce from hemocytoblasts, which ultimately, as we said, either going to form myeloid or lymphoid stem cells. So same deal, right? They're coming from, most of them are coming from myeloid, but one pathway going to lymphoid stem cells. And this myeloid stem cell, again, produce all white blood cells except lymphocytes. Lymphoid stem cell go through what's called erythropoiesis, which is the production of lymphocytes. White blood cells, except for monocytes, develop in bone marrow. Monocytes can develop into macrophages and other peripheral tissues as they go through their development. Now, we talked about EPO, right, erythropoietin. That's a hormone from the kidneys that stimulates red blood cell formation, right? Well, how do white blood cells? What is stimulating white blood cells to convert into varying white blood cells? And you have what are called colony-stimulating factors, or CSF. So those are basically the equivalent of EPO for red blood cells. You have CSFs for white blood cells. And they're hormones that regulate white blood cell populations. So you have MCSF, GCSF, GM-CSF, and multi-CSF. MCSF is going to stimulate monocytes. GCSF is going to stimulate granulocytes, which are neutrophils, eosinophils, and basophils. GM-CSF is going to stimulate granulocytes and monocytes. And then multi is really just stimulating everything, right? So if we look at, I guess we need to go back here. We look at this slide again, right? It may not be on this slide, but here we are, GM-CSF, GCSF, MCSF, right? It's what's driving the formation of these different white blood cell types, all right? So make sure you have a good understanding of CSFs, right? And then here's a nice snapshot of all the different white blood cells, how much you have, the appearance, what their functions are, other information. So this is a really nice way to review all the 5 different white blood cells. Neutrophils, eosinophils, basophils, monocytes, lymphocytes, and a lot of nice information about them there. So that's a nice sort of summary of those different slides. Platelets, as I said, are involved in cell fragmentation that is basically part of the human clotting system. Typically they circulate for about 9, 10, 11 days and then are removed by the spleen. Spleen reserves a lot of platelets in case of emergency if you have some sort of injury, so a lot of platelets are found in the spleen. Here you can look and see the different counts of platelets. The functions of platelets, again, really all are related to clotting, right? They release important clotting chemicals, right? Because the chemicals are released that attracts more clotting factors and other proteins to help, heal an area and repair an area. So that's a really important function. They temporarily patch or damage the vessel wall until this positive feedback loop sets in, and they can also reduce the size and the break of the wall. Platelet production also has hormonal controls in these 3 components here, multi-CSF, interleukin, and TPO. And then so here's a nice summary again of platelets. How much you're seeing, the appearance, functions, remarks, etc. All right? Now, hemostasis is the process of eliminating a cut or bleeding, right? That's a really important function of your body, right? If you have a cut or something, not going to be a good thing if it just bleeds indefinitely, right? So you need to eliminate this bleeding process, and there are basically 3 phases of what we call hemostasis. You have a vascular phase, a platelets phase, and then a coagulation phase. During the vascular phase, you have a damage to the vessel, right? Now, of course, if the vessel's damaged, you're going to get injury. You may get some vascular spasm as well, as there is smooth muscle around these vessels, and when the smooth muscle is impacted, right, it's going to constrict, right, and it's going to squeeze down, which can help maybe limit some of the bleeding that may occur in that vascular phase. The platelet phase is, as we just talked about before, aggregation of these platelets. Platelets can get off chemicals, as we said. Chemicals can attract more cells to the site, and that's what initiates this positive feedback loop. And then finally you have what is called the coagulation phase, where you have other clotting factors and other things that initiate this positive feedback loop, and the clot becomes stronger and stronger and stronger over time. So you don't need to know all of these individual clotting factors, for instance, that are listed here, but I do want you to have a general snapshot of what's happening in each of the different phases of coagulation or of hemostasis, right? 1, 2, and 3. All right? All right, so that ends our presentation here for the blood. The next one will be the heart, and I'll get working on that. Hopefully have it up, in a week or so. I try to do 1 a week, something like that. So, and of course, I'll also be seeing you guys in labs. If you have questions about things, we can always go over lab at that time. All right, so we'll see you soon.